preservation of and increased access to the 92nd Street Y Humanities Audio Archives is generously funded by the National Endowment for the Humanities.
Hello, good evening. My name is Susan Engel, and I direct the Adult Division in the Center for Adult Life and Learning here at the 92nd Street Y. Welcome to the opening of our Human Mind series, which continues for the next few weeks with some fascinating speakers addressing different subjects on the mind. Next week, John Kabat-Zinn will speak on mindfulness meditation. And the following week features cell biologist and psychologist Joan Borisenko discussing spiritual optimism. Also of interest to some of you, there's an addition to our program this year on Sunday evening, I think it's Tuesday evening, May 3rd. Anna Quinlan will return to the Y. She's the columnist with the New York Times, to be interviewed by Charlie Rose. And you've received a brochure of all our lectures, and I'd encourage you to buy tickets in advance to ensure your admission. And now, it's my pleasure to introduce the chairwoman of the American Committee for the Weizmann Institute of Science, who has graciously consented to say a few words of introduction for our speaker. She is Sarah Lee Schupf, familiar to everyone with a taste for delicious cheesecake. There actually is a link between the cakes and the Weizmann Institute. Sarah Lee's father, Charles W. Lubin of Chicago, was a baker committed to improving the world. He envisioned the impact of science on foods and chose the Weizmann Institute to establish the Lubin Center for Plant Science. Sarah Lee's own devotion to the Institute bears a personal stamp. She is a member of the International Board of Governors, chairs the American Committee, and has become a knowledgeable and distinguished spokeswoman for the Weizmann Institute. Ladies and gentlemen, please join me in giving a very warm welcome to Sarah Lee Schupf.
With that introduction, I feel like I should be doing more than what I'm doing. But I must say that the saying about nobody doesn't like Sarah Lee, really, once you leave here, you're going to realize why nobody doesn't like Weizmann. Founded 60 years ago as a modest research center in what was still Palestine, the Weizmann Institute of Science has flourished beyond all expectations. Its campus is the home of 2,300 scientists, technicians, and graduate students. Its researchers are valued around the world for the scope and impact of their contributions, from cancer research to plant science, the environment, and some truly spectacular mathematics. As you leave tonight, we will have brochures giving you information about Weizmann and its fine scientists. One of the most dramatic current areas of emphasis is the Brain Research Center. Yadin Dudai, our lecturer tonight, is one of its absolute superstars. You're in for a real treat. Professor Dudai is Dean of the Faculty of Biology, a native Israeli who earned his doctorate at the Weizmann Institute in 1974. He holds the Sella Professorial Chair in Neurobiology. Fellowships and research projects have taken him to Caltech, NIH, Yale, Columbia, and various European centers of science. Professor Dudai's focus is state-of-the-art research on the neurobiology of learning and memory. His topic tonight is memories, how we remember, and why we forget. I now present to you Professor Dudai.
Thank you very much. I hardly recognize myself in the introduction, and thank you, sir. I would like to share with you tonight some developments in the field of memory research. I will try to do it in a sort of a semi-non-technical language, in a way that will be understood by people who are not in the field. So don't worry if you see from time to time some technical terms, I'll try to explain them. And I will try to adhere to one single question, which related to a major subfield of memory research, which is the transformation of short into long-term memory. So the title of my talk is Memories, How We Remember and Why We Forget, or a journey into the mechanisms that transform short into long-term memory in our brain. I think the best way to start is to lead you from a behavioral observation via the anatomical parts of the brain to the chemistry. And the best way to start that is probably to perform an experiment on you. You see, I'm an experimental scientist, and I believe in data, and I must utilize this opportunity to collect some more. So I would appreciate it if I get some like 3 or 4 volunteers from the audience. It's not very painful, and I can use the Israeli way, which is I volunteer you, and are there other people here that volunteer? No, you can sit in your chair. It's 2, 3, 4. It's enough. Okay, now I'm going to perform a very simple experiment with a sort of a very deep morale. And I am quite sure that some of you are familiar with this experiment. As I'll tell you later, this experiment actually opened the era of modern research on the psychology of learning. And what I'm going to show you is an array of random numbers on the screen. It's not going to be performed in a strict scientific manner, but just as a demonstration. I would appreciate it if the volunteers, and only the volunteers, raise their hands and they don't do the following. They don't write the numbers down when I show you. They don't write them down, thank you. They try to remember them. They don't shout the numbers. They just raise their hands. And I would really appreciate it if other people in the audience, they can obviously remember the numbers, but they don't shout it and they don't interfere in the experiment. So here we start. First one. Okay, volunteers. You. Same. Okay, I don't remember the numbers myself, so I have them here. And since I usually forget where I put my papers, I have them twice. So just to be on the safe side. So as I see here, the number is correct, okay? Now let's proceed. Volunteers, you. Do you agree? Do you agree? Try to repeat the number. 5. Thank you. There is a problem with the experiment. Okay, so it's a slide projector. Okay. Anybody disagree?
47, 20, 53, 18.
Okay, I'll tell you the truth afterwards. Okay, I'm going to proceed to another one. Please don't write it down. Volunteers. At that stage, I'll accept volunteers from the audience as well. Who wants to try? Okay.
438, 650, 431, 79.
Somebody else? Okay, I have the advantage. I have the numbers here. Well, I'm happy to inform you that you are all normal. If you perform such an experiment on a normal population, on a sample of a normal population, and you present a population with an array of random numbers or random letters or random bits of information. I'm not going to define what a bit of information is for that purpose. For our purpose, it's a random number, okay, not a random digit. And you present the information for a while, let us say for 5 seconds to 10 seconds. And again, I didn't do the experiment in the right way because I didn't measure the time. And then you plot the number of correct responses here versus the number of either dots or letters or digits that you are presented. You get the following pattern. Usually, you get 100% response up to number 7, which means if you have up to 7 random bits of information, and then the number of correct responses declines, and around 11, it approaches 0. So what I did here is to present you with, when I reached 8 digits, you were still okay. 3 out of the 4 were okay, which is rather good. And when I reached 11, well, you didn't remember, and that's normal. This kind of experiment was designed by the founding father of modern psychology. His name was Hermann Ebbinghaus in the 1880s. And the reason he devised this experiment was, first of all, he wanted to quantify the research on memory. He wanted some quantity to measure. That's the only way to convert a field into science. And second, he didn't have grants, so he had to do all the experiments on himself. So by doing that, he opened the field of psychology and found 2 very interesting phenomena. One, we found ourselves a minute ago, and it is the capacity of our memory is rather limited. If we go on above 7 random digits or random letters, we start to forget. This is a very limited capacity. So how come that we remember so many things? The answer is, so this is the outcome of his experiment, and this experiment can be repeated in various manners, various forms. The capacity of short-term memory in our brain is limited. We can store only 7 bits of information at a time, and there is actually a known sentence, a known saying in psychology, which says, this is the magic number 7, or we call it better, the magic number 7 plus minus 2. If you remember less than 5 random bits of information, you may have a problem in your memory. If you remember more than 9, please send me your name, and I'll be happy to conduct some experiments on you. Now, how can we remember? By the way, I must add something else. I can remember some of these numbers, and I hope you will forget them. Because these numbers are my ID number, the number of my Visa card, and that's the only way I can remember them, because they have some importance to me. How come we remember so many things if the capacity of our short-term memory is so limited? Well, the answer actually lies in the question. I ask, how come we remember so many things when the capacity of our short-term memory is so limited? We can remember because there is a transformation of information from short into long-term memory. And I can tell you another outcome of this kind of experiment, and I'm not going to repeat this type of experiment on you. If I would present you each of these digits or letters for more than 10 seconds, so for example, for 11 numbers, I'll expose it for more than 2 minutes, you are going probably to remember them for longer, because it takes between 10 and 30 seconds to transform normally one such bit of information from short into long-term memory. So we have in our brain at least 2 types of memories. One of them is a short-term memory, which you use in everyday life for such tasks as remembering a phone number that you have to dial, and remembering the license number of a car that just drove past you in the red light, or things like that. And we have the long-term memory, which is keeping the information which is important for us. I remember these numbers because they are important for me. They mean something to me. So if the information is meaningful, you keep it. So this is a behavioral observation, a very simple experiment that shows you that we have short-term memory and that the capacity of short-term memory is limited. And if we have short-term memory, we have also long-term memory. And I would like to lead you throughout this lecture in the coming 40 minutes throughout various levels of organization of the brain and try to explain to you, at least provide you with a partial heuristic explanation. How come that the brain takes this information from short into long-term stores, put it into long-term stores, and what happens in the brain when these events take place? So I started with the behavior. I'll go through anatomy, which means parts of the brain, and they'll end up in molecules or chemistry, and if you wish, pharmacology. And I hope by the time I end my lecture, you have some additional insight into a process that it's so easy to follow from a behavioral point of view. So one thing I promise, I'm not going to conduct more experiments on you. You can relax, and I'll proceed. So some memories are retained for only seconds to minutes, and we define them operationally as short-term memories. And some memories are retained for months to years, even to a lifetime, and we define them operationally as long-term memory. And there are two conditions that underlie the transformation of information from short into long-term memory. One of them, information is being transferred from short into long-term memory if the information is intensive or rehearsed. If I give you enough time, you rehearse the information. If it's important to you, it will go into long-term memory. Another condition which might fulfill the same role from an anatomical or neuronal point of view is if the context signifies, either explicitly or implicitly, great importance. If the information is very important to you, even after a single trial learning, you may remember that. Okay? So we may try later on to find out what is the meaning of these constraints from an anatomical and molecular point of view. So again, it's a behavioral explanation, but we would like to see what is the meaning of these mechanisms when they operate in the brain. So the question I would like to pose is the general question that covers the entire lecture. What happens in the brain when short-term memory is transformed into long-term memory? And the way to start is to start with a certain level of organization of the brain. Because you see, whenever we study the brain, we have to decide at what level of organization we proceed. The brain can be studied at a behavioral output level. That's just what we did a few minutes ago. We studied the input-output relationship. The brain can be studied from an anatomical point of view. I can dissect the brain and explain what each of the parts is doing. The brain can be studied from a molecular point of view, the chemicals that operate inside the brain. So I will start with the anatomical level and give you some very brief, somewhat, I am afraid, superficial, but I think sufficient for our purpose, overview of one constraint that will explain why memory is transformed under certain conditions into a long-term store, and other conditions this process fails. Before proceeding, I have to define more clearly to what kind of memory do I refer in my talk. And here I must explain a bit more. You see, one question you may ask yourself, and maybe some people in the audience already did it, is how many types of memory do we have? Do we have only one memory store, or do we have several of them? It's very easy to show you using very simple self-conducted experiments that we have more than one type of memory. For example, I may ask you a question like, what is the capital of France? And you would say, Paris. And this information is shared by people in the audience because they have learned it either in school, or they watch TV, the news on TV, or they read it in the newspaper, or they heard it from friends. This is a bit of information that we call semantic information, semantic because it's usually conveyed by language. And this is information that can be shared by many different people in the culture. So many people here in the audience may share large chunks of semantic information, like what is the capital of France, where is the Weizmann Institute, and other important questions. Okay? Now, there is another kind of memory which is based on another type of information. I expect many people in this audience to have been to Paris. Some of you have been there, I'm sure. Those who have been there, would you kindly raise their hands? Okay. It's easier to ask how many people are here haven't been there, but I think So if I ask you people, those who have been there, what is the capital of France? For you, the answer contains some autobiographical elements, because you may remember what happened to you in Paris, okay? Now, this kind of information is not merely a fact or a semantic type of information, it is also an autobiographical type of information. Now, please observe the following fact. Many people in this audience may have large chunks of semantic information which are similar to semantic information that is contained in the brain of the people who sit adjacent to them. But there are 4.5 milliard brains on this planet. At least this is the number I got yesterday. None of them contains the same autobiographical memory. None of them. Autobiographical is unique. Autobiographical memory is unique. So we have two types of memories. One of them is a memory which is semantic, another one which is autobiographical. What is shared by both these types of memories? In both cases, when I ask you the question, you reply, Paris, in that example, you are aware of the fact that you provide the answer. You sort of declare the information being aware of what you say, what you utter. This is called declarative memory, and autobiographical and semantic memories are both subtypes of declarative memory, memories of which we are aware, and we can, when we enter the answers to the questions, we know that we provide information. There is another type of memory, and by that the end, because I end that, because I can continue and divide it at least 6 or 7-fold more, but for our purpose it's enough. There is another type of memory. If you, for example, own a car, and you drive your car from your home to work, and you arrive at your work, and I ask you then, can you tell me how many times you press the brake pedal, or how many times you turn left or turn right? You're bound not to remember that. You cannot provide me with the answer unless you are a novice driver. The reason is that you use information, but you are not aware of the fact that you use it. We call this type of information procedural or automatic memory. Lots of the information that we use in our daily life is automatic. It's not declarative. Now, why did I say all that? Because I want to show you one region of the brain which is critical in the consolidation of short into long-term memory, which is related only to declarative memories and not to procedural ones. And I had to make this distinction in order to provide you with the correct answer. I want to refer only to one subtype of memory, the memory which is declarative. For us, the memory which seems more important because it contains the autobiographical episodes and the semantic facts. Now, in the case of declarative memory, so I just said that there are several types of human memory. One of the common classifications, there are other types of classifications, distinguishes between declarative and procedural. Declarative is about facts and events, and procedural memory is about procedures. So we are going to forget about the procedures and stay with the facts and events, declarative memory. Now, declarative memory, so says the current conceptual framework, is stored in that part of the brain which is called the cortex. This is a cross-section, in this case, of a monkey brain, okay? But it's very similar to human brain. Sometimes it's very difficult to distinguish. So if you take this part of the brain here, it's called the cortex. It is the physical substrate of the human, most of which, what we regard as the pinnacle of the human mind or human culture. The cortex is divided into sub-areas, each of them spatializing information that comes from a specific sensory modality, like visual information, auditory information, taste information, somatosensory information, touch, and so on. And we now believe that the information that is stored about these modalities, information derived from this via these modalities, is stored at various areas in the cortex in a way of a distributive manner, which means it's not stored in a very specific location, but in an area which uncovers that area which deals with those information that come from that modality. However, there is an additional region of the brain, additional part of the brain, which seems to be extremely important in keeping memories stored in the brain, or better, in consolidating short into long-term memory. And this is a subcortical part, which we call, the scientific term is the limbic system. Now, you should, those of you who are physicians or are familiar with anatomy know well that many of the names of parts in the brain are derived from their shape, because people, when they describe them first, didn't know what they are doing. As a matter of fact, right up to now, we don't know what many of these parts are doing. So the limbic system is that part of the brain which is on the border of the cortex, and limbic means border. The limbic system is a subcortical area, and it is shown here in colors. It's subdivided into various parts, and you don't have to remember the names of this part. Few of my students remember, okay? All you have to know that there is a part of the brain which is subcortical, and it's called the limbic system. For example, one major part is called the hippocampus. You may have seen this name here and there. Now, it appears that in order for memories to be transformed from short into long-term memory, which means to be deposited in the cortex, the limbic system must be active in a normal manner. If you damage the limbic system, you get a stage where memories cannot anymore be stored. This is called amnesia. But amnesia only for declarative memory, not for procedural. That's why I made the distinction. Damage in the limbic system will prevent memories from being consolidated from short into long-term memory. By the way, this is exactly why many inflictions in the brain cause amnesia. For example, strokes in the limbic system, Alzheimer's disease, some inflammations, strokes in this part, Korsakoff syndrome. All these diseases or inflictions affect the limbic system. So even if the cortex is not affected, and it seems like memories can be retained there, memory is not being transformed from short into the long term, because the limbic system must somehow be activated. So a part of the brain which is termed the limbic system is critical for the consolidation of memory into its long-term form. Damage to the limbic system may result in inability to remember, amnesia. And people with a severe damage to the limbic system, and there are cases like that, cannot remember or cannot acquire information, cannot learn anything new which is declarative, which means memories that they are aware of. The damage usually does not affect memories that are already stored in the cortex. It affects only, usually, new memories. So, for example, if a person at 20 years age is lesioned in his limbic system, and the lesion is severe enough, you may end up with a person who is 60 years of age, after 40 years, he still remembers very well, or reasonably well, what happened until the age of 20, when the lesion took place, but afterwards, he didn't learn anything new, which is declarative. I make this distinction because they can learn procedural memories, and this is another story. I don't want to get into it. So the bottom line is that the limbic system is, from an anatomical point of view, part of the flow chart of the system that is needed to store, is needed in order to convert short into long-term memory. When I gave you this test, I activated your limbic system. If I were to repeat the numbers enough times, your limbic system would be activated and transform the information into part of the cortex. And if it's strong enough, the trace is strong enough, it will be stored there for days, for weeks, maybe even for years. By the way, any of the volunteers wants to volunteer the first number? 4729? No. Volunteers. There's another damage to the limbic system that causes people to talk with them. Okay, you come to me afterwards. The number was 4927, and actually it's entirely normal. You didn't have time to rehearse. I turned your attention to something else. How come you remember even the numbers, not the order. You see, what I did was to turn your attention to something else, because I started talking about some other things. Now, one additional system in the brain that is extremely important in storing information, or in my terms, consolidating it from the short into the long term, is the attention system. What is the meaning of the attention system? There are systems in the brain that are activated when we become attentive to something. When do we become attentive to something? Either the context is very important for us, or some very bizarre or surprising events take place. For example, if you drive your car, even using procedural memory, and you see the blue light flashing in the mirror. I don't know if it's blue here, it's blue in my place, okay? And this immediately will cause your attention. You may remember what you have done then, okay? So, there are additional systems in the brain that are critical for consolidation of memory. For example, hormonal systems that encode contextual importance. When you see the policeman, some hormonal systems in your brain start being activated, which is entirely normal. Damage to this system may also hamper memory. I want to give you only one example. This is an example, a very schematic example, which depicts the human brain. Information comes via the eyes, which are supposed to be here, and it goes via several stations to the cortex that deals with vision. It's called the visual cortex. Now, the information follows the blue path here. There is a system in our brain which is in the basal forebrain. This is another term you don't have to remember, just remember it's somewhere there, which is here depicted in red, which releases a transmitter, a chemical that transmits information, which is called acetylcholine. Acetylcholine is rather famous chemical, and I think it's mentioned in the New York Times once in a while. So it's a very simple chemical and very important. By the way, it's important also because it's related somehow to the damage caused in Alzheimer's disease. But we are not going to talk about that now. Now, this system here signifies information, that signifies importance or contextual importance. Something important happened. And it sends information to the visual cortex. Only when these two events arrive together will we remember that information. So you didn't remember the numbers, although I admire the fact that you remember the numbers, not the order, because I didn't let you activate your attention, okay? So here is another anatomical system that is important for remembering. So all I want you to remember from that part of the talk is, first of all, from the first part, there is short-term memory and long-term memory. You transform short into long-term memory under certain conditions. This transformation depends on at least two types of systems in the brain. One of them, the limbic system, which is a sort of the gate or the central processor, and the other one, those systems that signify attention. If any of this is damaged, you are bound not to remember. So, just a second. So, you may have people, for example, people who are amnesic for declarative memory, have a damaged limbic system. They still remember short-term events. They may remember up to a minute, even 2 minutes, and then they forget. The limbic system is not activated. Some people with damage to the cholinergic, cholinergic means the system that synthesizes acetylcholine, the transmitter, can remember events for a few minutes, but not for longer. I suggest we defer the questions to the end, okay? If you remember that question by then it's important. This is called selective memory. Okay, now I want to proceed further and ask what happens at the molecular level. So I gave you some hints about things that happen at the anatomical level, and I want to ask what happens at the molecular level. Now I want to start by saying I'm going to show you some slides which contain chemical terms. Don't be offended by them. You don't have to remember the terms. You have just, if you bear with me, you just follow the rules or the concepts, and that's enough. I have to confess that exactly as I don't remember these numbers, I myself don't remember many of the chemical terms. I know where to locate them. I know what these chemicals do. Okay, so you have to remember only part of them. So let's proceed and ask the question, what happens at the molecular level? In order to do that, I want to sort of use a metaphor in order to explain what do we think happens in each of these stations in the brain when memory is stored. Well, each of the parts that we mentioned contains millions or zillions of neurons. Neurons are nerve cells. And information is stored in the activity of these nerve cells. And it is stored in the temporal and spatial patterns of activity. Okay? Now, instead of depicting a network which is 10 billion neurons, I depict here a network which is only 4 or 5. Okay? So each of these is a nerve cell, and information flows from one cell to another. So we have a circuit. And in learning and memory, changes take place in the efficacy of information transfer or in the connectivity of this circuit. So the connectivity in the circuit changes, and due to that, the representation of information in that circuit changes. If you wish to use a metaphor, the easiest one is to remember this beautiful picture. Okay? Suppose each of these is a nerve cell. Okay? The handshaking is the connectivity. Memory means that you change either the strength or the order of the handshaking. So the major events take place here. In this case here, for example, the hands do not yet touch. When memory takes place, these hands will become connected, okay? The major events take place in these connection regions here. So here, connections here. These points here I call synapses. Synapses are functional connections between nerve cells. So these are synapses. Here, here, here. And this is a non-functional synapse because it's not yet connected. So events take place there. And the chemical events I'm going to describe take place in synapses. That's all we have to remember. Now, this is a very complex slide, and I'm not going to go into it, not because it's complex, but because we don't need the detailed information. Only one thing I want to emphasize. This is a schematic, highly simplified depiction of a synapse. Information arrives from the presynaptic terminal, which is here. See here? This is presynaptic. Information comes, for example, from the eye, from here, and goes to the postsynaptic, to the next cell in the line. And in between, there is a gap. And information flows from here to here via chemical messages. These chemical messages are the secret of the connectivity of the nervous system. By the way, most of the drugs that we take act by interfering with these chemical messages. And I'm not going to go into details. But those are the clues to many of the events that take place in the nervous system. All I want to say is that when short-term memory takes place, or when we acquire information, some changes occur in this synapse that cause changes in proteins. Proteins are the major constituents of the body, and as such, they are the major constituents of nerve cells. And when they change, some of these proteins may alter the efficacy of information transfer in the synapse. So without going into details, all we have to remember is that there is a change in proteins when the information comes during learning. Let me give you another way of looking at it. Suppose I watch you. Information arrives from the eye and goes into the brain. This information travels along the nerve cells as an electric current. It arrives at the synapse, releases chemicals, and they continue the information transfer in the second cell and next cell in the line, and so on and so on and so on. This chemical information causes alterations in the proteins of the synapse through which it goes. And these changes in the proteins, which are depicted here in various forms, are changes that lead to alterations, if you wish to use electrical terms, in the resistance of the membrane, or if you wish to do it to use other terms, in the efficacy of the transformation of the information along the nerve cell. Or if you wish to use another term, you may resemble the synapse to a wire, and now the wire becomes wider. Information flows easier there. The only thing we have to remember, without going into details, because we know a lot of details about those events, is that there are alterations in proteins. That's the only thing we have to remember for now. Now, in short-term memory, the novel information alters the structure of existing proteins in synapses. Remember, synapses are the functional connections between the nerve cells, and hence altering the efficacy of information processing in the circuits. So without going into details, there are changes in proteins, like various pores or various enzymes that digest proteins and so on and so on and so on, and the connectivity changes. This is the major effect of learned information, and short-term memory is the change in the structure of the proteins in the synapse. Short-term memory is the alteration in the proteins in the synapse. Information is retained by the retention of these changes. In a minute, you'll see why it's important. Now, there is one critical point that we have to remember. Proteins do not live forever. The half-life of a protein in our body is between several seconds, usually minutes, to several days, weeks at most. This is the way we are in a constant turnover in our body. Every minute, lots of proteins in our body are destroyed and new ones replace them and so on and so on. We are in a constant change, okay? Now, why is it important? Because I just said that short-term memory is based on alteration in proteins, existing proteins. If this is the case, and we know this is the case, then there is some problem. If memory is based on alterations of proteins, and proteins live, the lifespan of proteins is only minutes, maybe hours, maybe days, we remember some things for years. So how come? How come it happens? Remember, short-term memory is subserved by alterations in existing proteins. Proteins don't live forever. They have a very short half-life in terms of memory spans. So how can we retain the memory? This was a question that haunted many researchers in the field for many years. Here is one possible answer. So remember the dilemma. Short-term memory is subserved by alteration in existing proteins. Proteins have a very limited lifespan. Memories live much longer. Here is a scheme that depicts one possibility. Actually, you see here 3 cartoons of neurons. This is a cell body and this is the synapse. Just a very simplified cartoon. Here you see the time course of memory. Suppose I have a stimulus that is remembered, and this is the lifetime of the memory. This is the lifespan of the memory. As you see here in arbitrary units, the memory decays. The reason for the decay is that the memory is based on alteration in a protein. The alteration is here depicted as the green dot, and this is destroyed after a while because the protein is destroyed and the protein is remodified. Memory decays because the change either relaxes back to normal or is destroyed. One possibility that I would like to earn your attention is please skip B, which is not important for our purpose now, and go to C. What we know now is that in long-term memory, a very unique and exciting event takes place in the nerve cell. The information, if it is sufficiently intensive, or if it is in the context of additional information, remember from the behavioral point of view, either we rehearse the information or something important happened, the information now not only transforms proteins into new phase, into new stage of that protein, not only modifies the protein. By the way, the scientific term for modification of existing protein is post-translational modifications. For those in the audience who are familiar with a bit with biology, post-translational modifications. We changed existing protein. One possibility is now that the protein goes into the nucleus of the cell and does something else. It doesn't go directly. The change causes other changes that go and modify something in the nucleus of the cell. Why is it important? Because the nucleus of the cell contains nucleic acids. Nucleic acids are the genetic code. What the long-term memory process does or is subserved by is an alteration or modulation of the expression of the genetic code in that nerve cell. Why is it important? Well, first of all, now you don't rely on modification of existing copies of protein molecules because you modify genes. In a way, you modify the expression of genes in a way that will cause now new copies of the protein to be produced constantly. So memory can stay there for as long as the gene is active. So we overcome the problem of the limited lifespan of the protein. We call it, we render the system immune to molecular turnover. But here is another point. In consolidation of long-term memory, the novel information modifies the expression of genes in the nerve cell that continuously produce new proteins. This renders the system immune to molecular turnover. And this turnover is the fact that limits the duration of short-term memory, I just told you. I'll show you one example. Don't be alarmed by the fact that this contains various symbols. It's very simple. This is again a cartoon of a nerve cell. This is the synapse, this is the nucleus. As you see, nerve cells come in various shapes. And this is the teaching stimulus. Information, for example, from the visual system, auditory system, and so on. Short-term memory is based on modification of proteins, which are here depicted as the yellow boxes, various types of proteins. Long-term memory involves the modulation of genes which are in the nucleus. In both cases, the products of this modulation will cause changes in the release of the transmitter or in the efficacy of the information transfer in the nerve cell. We know a lot about these processes now. We know quite a lot, although we don't know enough. We can describe them in great detail. We identify molecules that are involved, processes that are involved. And this is a very fascinating field. This is a very, and the last complicated cartoon, this is a very simplified version of only a small part of the events that take place in a nerve cell when information is being transformed from short into long-term memory. And what you see here is interaction with the cell surface of the information that comes from the synapse, activation of certain types of genes in the cytoplasm, which means outside the nucleus, and then information goes into the nucleus, and all these seemingly complex events are actually only a small, tiny fraction of the events that take place in modulation of the expression of genes. So what happens now in the nerve cell when information is transformed from short into long-term memory is that the genes now will produce new arrays of proteins, which will cause the modification of information transfer. By the way, for those in the audience who are familiar with biology, these molecules here are actually subtypes of oncogenes. Oncogenes are genes that cause malignancy. But this is another story, and it just alludes to the fact that these mechanisms are shared by very different processes in the body. Why do I say all that? Because I have a moral in that, and this is the following. The molecular mechanisms of consolidation of long-term memory resemble those of tissue differentiation and development. What happens when in our body the tissues develop, either differentiate or develop? The same thing. Information comes to a cell, but in this case not necessarily a nerve cell. The information does something to proteins. If the information is in the right intensity or the right permutation of signals, it will cause modulation of gene expression in this cell, and the modulation of the genes in the cell will modify the properties of the cell. So this is exactly what happens in development or differentiation in the body and other tissues. So what actually happens in consolidation of short-term into long-term memory is that the brain is continuously developing while we learn. So the most important point to remember is that when we learn information that is stored in the long-term stores in our brain develops continuously. And many people would say that the brain is the most immature organ in the body. And I'm not referring only to politicians. I'm referring to what's happening in the brain. So the brain is continuously developing. No, it does not get bigger. I'll explain in a second what it does. The brain is continuously developing, which means we get more and more connections in the network. We have a certain part of the brain that stays in a mature brain at a reasonably constant size, okay? But the richness of connections inside this region increases with time as the brain exercises, as the brain remembers more and more, which provides me with the answer to a question to which I am referred very often. Can we improve our memories? Well, we can if we keep rehearsing. How do we keep our memories going? The best way, keep using it. because whenever you use it, you provide novel substrate for novel information. If you don't use it, you don't exercise, it's exactly what happens to your muscles. So it's not a remedy. This happens only under normal conditions. But it's very likely that if you don't use your brain, you have a less complex network in those parts of the brain that are required to store information. And it's very easy to do experiments. Well, it's not very easy. It's relatively easy to do experiments on animals and to show that animals that grow up in an enriched environment, which means environment that contains their peers, some toys, stimuli, will have a richer, denser parts of the brain as compared to those that grew up in a non-enriched environment, which means either in isolation with no stimuli and whatsoever. This, we think now, happens also in a mature brain. So the bottom line is, if you want to keep your memories going, which means if you want to enable your brain to store long-term memories, keep using it. So what I described today is research, or actually very tiny bits of the output of research that took place on 3 levels of organization of the brain. The behavioral level. I've shown you that there is short-term and long-term information, and I wonder whether you still remember now the numbers that I said. Do you remember the first number? I don't. What is the first number? 4, 9? Okay, because I turned your attention to it. That's the only reason. I turned your attention to it. You rehearsed it. That's why I remember it. Is there anybody here that remembers the second one? 64,000 shekel question. Please. Could you kindly repeat? No, the 5 is correct. You see, I have the data, so that's the only advantage I have. Now, so I showed you on the behavioral level, there is short and long-term memory. I tried to explain that there are circuits or parts of the brain that are required for the transformation of short into long-term memory, and gave you 2 examples. One is the limbic system, which is a sort of a central processor, and the other one is the system that signifies attention. And one of the example I brought is the acetylcholine system, which, as I said, is damaged in Alzheimer's disease. And then I told you about what happens in cells and molecules. And I told you that in short-term memory, the system uses modification in existing proteins which have a limited lifespan. And long-term memory involves, as far as we understand now, and this may change because in science the picture changes every few years. That's the thrill. The information involves transformation of data, of signals from the cytoplasm to the nucleus, modulation of genes, which causes the system to remember much longer. And I ended up by saying that the best way to keep memory going is to use it. Thank you very much. And I said that I'm supposed to take questions from the audience? Both people here say yes, so. Okay, I'll start with you, please. Yeah, I can use this, but this is not very healthy, so.
Why is it that many older people who do not have Alzheimer's have problems with their memory?
Okay, first of all, the question was, why is it that many older people that do not have Alzheimer's seem to, I add the seem to, have problems in the memory? Now, first of all, I would like to emphasize I'm not a clinician, so I am bringing you information from the research field and not from the clinical point of view, although we do work together with clinicians and neurologists especially. One must distinguish between 2 phenomena. One of them, it is rather clear that with age there is some normal deterioration in the capacity of the memory system. However, we must distinguish between the inability to store the information and the inability to retrieve it. It very often happens that we find it difficult to remember, but we remember later. We can retrieve the information, it is there. So many of the problems are due to retrieval problems. Retrieval problems are due to two factors. One of them we don't know. And the other one is problems with attention. We find it very difficult to retrieve when we are not attentive. Now, attention problems derive from both two facts as well. One of them you know that we don't know. And the other one is anxiety. Anxiety. You see, a large proportion of people who present themselves to clinicians with memory problems, apparent memory problems, and I hope that there are clinicians in the audience to support my view. I have the data from Israeli hospitals, I assume that's the same in the United States, are people with depression. They usually don't have memory problems, they have problems in retrieving the information due to the depression, which probably is accompanied by some anxiety. So if you feel that you lose your memory, it may exacerbate the problem. Now, we don't know what are the other factors, but one must distinguish between the two. So I'm not claiming that there are no problems of memory with age. I used to say that they start after the age of 20, and then after the age of 30, after the age of 40, 50, and so on, but they do occur. But we don't really know now whether they are due, in the majority of cases, to retrieval problems or to storage problems, and we know that many of them are due to retrieval problems. Oh my gosh, please. I have a very short-term memory capacity, so I remember only you. Okay.
Where does the eidetic memory scheme fit into your explanation?
Whereas the eidetic memory scheme fits into my explanation, I would like to add a word to that. The classification of memories that I brought was a very highly simplified one, and referred only was used here to subserve my wish to bring to your attention the existence of the limbic system. There are memories memories can be classified also on the basis of the time scale, for example. We have sensory information. We have sensory information that we can keep for, let's say, tenths of seconds and not more than that. We have information that we can keep only for fractions of a second. We have information that we can keep and we can classify on the basis of the sensory percept that caused this memory. So I didn't have time to go into it. The only thing I want to mention, to emphasize, is all these various types of memories very probably use the same or similar, not the same, similar cellular and chemical mechanisms, only they differ in the place in the brain in which the events take place. So the major point to remember is that if you are dealing with memory which is short-lived, or memory that is subserved only by a certain sensory modality, you probably use the same mechanism described by me. So I didn't have time to go into various types of memories. I just refer to a single one. The question was, genetic material is carried to the offsprings. If memory, long-term memory, involves modification of genes, I use the term modification, you'll see in a minute why, can we carry the memory from one generation to another? So I have a very important question. Is this fresh water? Okay. Thank you. Now, the important term that I used was modulation, and not modification. I want to explain why. Almost all the cells in our body carry the same amount, the same pattern of genetic material. So potentially they can express various genes. The genes that are expressed in a certain tissue are determined by the way that the genetic material in that tissue is regulated. When I spoke about modulation of genes, I meant not changes in the genetic material, but in the expression of the genetic material. Genetic material is transformed from one generation to another. It will be transformed also from that generation to the other one, but not the modulation. So I don't change the gene. I change only the expression of the gene. Is this clear? Okay. Suppose I have here, may I still use the slides? No, okay, they're already here. So I can use them, but not for that. Suppose I have a gene for an enzyme, like alcohol dehydrogenase, an enzyme that does something to alcohol, okay? And this gene in a certain tissue is not expressed, which means the gene is there, but it is not, the genetic material is not translated into the protein because there is a block on that site on the genetic material which is required for the gene to be translated. Is this clear up to now? There is a block there. I can modulate the expression by removing the block. I will not change the gene. I will just allow the gene to be translated. This is what happens in long-term memory. The gene is not modified. It is just that the regulation changes. Now, from one generation to another, I transform the genes, but not their regulation. So, therefore, long-term memories are not transformed from one generation to another. It's only the capacity to learn, not the memories themselves. Capacity, in general terms, is transmitted, but with a very wide margin, which means you have a certain capacity to learn, but the way this capacity is materialized is determined by the environment, especially in the first years of life. There at the end. I can go back to number 7.
All the billions of neurons, why in the short-term memory do we have that dramatic fall-off
Yeah.
between the 7 and the 11?
Tell you the truth, we don't know the answer. But I would like to turn your attention to the fact that this is not due to a limitation in the number of neurons, but the limitation of the operation of the system at the flowchart level, the operation of the system. So we don't know exactly why. We can understand that it imposes, it is sort of having a large short-term memory capacity is not necessarily important for survival. You might have, but this is a teleological explanation. So I might explain to you why do we have it, because you need a lot of energy probably to keep this amount of memory in the short term, and you don't need more for usual tasks. But we don't know the cellular or the anatomical answer for that. By the way, animals as primitive as insects have a capacity of about 4. But this is, of course, not linear, okay? Please.
Different our synapses are getting, as we get older, they decrease the connection between our brain. How is a long-term memory? Is it a result of intense feeling that we recall events even our synapses decreases?
No, what happens is, so the question was if the number of synapses, of connections in the brain decreases with age, and this was a presumption, an assumption you made, which I have to refer to, how come we still remember lots of events? That's the translation of the question? Okay, so first of all, the number of synapses probably decreases with age, above the age of, let us say, well, I would say 66, but it doesn't matter, probably it's much longer. Yeah, but probably the capacity of the system is so large that under normal conditions, for long-term memory, you wouldn't feel the effects. Now, you ask, how come a system, I will translate your question to another manner. How come that a system in which the connectivity decreases, a localized part of the system, still remembers? And the answer to that is that information in the brain, and I alluded to that at the beginning, is stored in a distributed manner, which means it's not stored at a certain connection, but in many connections altogether. And even if you lose some of them, the information still can be completed by the other parts of the system. We call this, in the scientific term, graceful degradation. If you take, no, it's a serious scientific term. If you take, and it's graceful, if you take a system that is composed of many units, and you degrade part of the system, you have to degrade a much larger part, a really large part of the system, in order to get the memory evaporate from there. You can still have memory stored with parts of the system destroyed. It's like having a puzzle with many repetitive elements. Okay? I will take 3 extra questions. You there. Okay, the question was, when a baby is born, is there a memory from previous generations? Okay, actually the answer is yes, there is genetic memory. You see, in the brain, we have built in the brain patterns of behavior which are coded by the genes. And babies can perform a lot of things without learning to perform them. Those are what in everyday life we use the term, which is misleading, instincts. The information is there, it is transformed from one generation to another. The only point to remember is that even that information must interact with the environment in order to develop. Okay? Now, other information is not transformed from one generation to another. So even if you sit in university and learn and you learn and you learn and you learn, your son or grandson will still have to do that again and again. 2 questions. There. There are vitamins on the market for memory. Are they helpful? It's a complex answer. About 15%, again, I would appreciate it if there are physicians in the audience that will support that, because again, it's Israeli data. About 15% of the complaints on memory above the age of 65 are due, 15, it's not a small fraction, are due to deficiencies in vitamin B. However, 85% are not. In addition to that, on the market, if you go to a drugstore, you find lots of preparations that promise you long life, a happy life, better memories, and so on and so on. I don't know whether this was determined in scientific experiments. The only thing I can say is that under certain conditions, deficiency in vitamin B will cause memory problems. We see that in the old, and we see that also in people who consume large amounts of alcohol. And this is the beginning of Korsakoff disease. But in normal conditions, people should have a normal supply of vitamin B. So I don't know what happens to a person who is normal in terms of his vitamin B supplies and he chews a lot of vitamin B. I know what happens to the guy who sells it, but I don't know what happens to the parent. Now, there are some factors which are required for memory. And at a certain age, when we don't eat properly, we may lose some of the stores. For example, certain fatty acids, like choline. Choline is important for the generation of acetylcholine, which I just mentioned, okay? But I don't know whether under normal conditions, if you chew choline, something will happen to you. Under certain conditions, diarrhea, but this is not related to them. So what I'm claiming is that the fact that under certain deficiencies, you must supply vitamins in order to improve your memory does not allude to the claim that under normal conditions, consumptions of these vitamins in megadoses will do anything. I have a personal view that the only foodstuff that improves memory is chocolate, but I'll debate it later. Last question, you. Can we stop here? Because if you go to the second one, I'll forget the first one. Okay, first question, and you remind me of the second one in a minute. First question, what's the role of the limbic system? This is a major question in modern research. What is the real role of the limbic system there? We think that the limbic system is used in order to strengthen the connections in those areas of the brain that later store the memory. So it's very similar in the sort of scheme that I showed you on the acetylcholine system. Information came from a certain point and acted on another point. So very probably the activity of the limbic system, which is influenced by our attention and so on, and excitation and so on and so on, has to come to a certain point in the cortex and strengthen the connection there. After a while, after the connections are strengthened there and the circuit is already facilitated, the limbic system is not needed anymore. So it's not part of the circuit. It's only a modifier. Yeah, activity goes in the cortex. What was the second question? The question was, are intelligence and memory connected together? I'll make it, because it's the same question. Now, in a way, yes, you must have a certain normal level of memory capacity in order to reach a certain normal score in memory, in intelligence test. I'm not defining intelligence here. It's a long topic. It's a very heavy topic. I don't want to define it. I'm just claiming that you, in order to reach a certain normal level of performance on the tests that are used nowadays in order to evaluate something which is called intelligence, you must have a normal level of memory. However, as far as I know, there is no direct relationship between supermemory and superintelligence. Very intelligent people may have rather lousy memories. So the connection is not very simple. On the other hand, there are people that might be called idiot savants, which means they can have excellent memories, but their intelligence scores are very low. So if you have a very good memory, relax, it doesn't say a lot. Thank you very much.
Thanks for listening. For more information on the 92nd Street Y, New York, and all of our programs, please visit us at 92ny.org.